You playing? Sure.
All right, I'm going to shut off the projector and I'll come back to this. Actually, you know what? Let's keep the projector up since I have it on. And let's go to a codon chart and let's do some codons.
And you'll give it.
Yes. I'll give you the chart.
Patrick, can you like slouch your head?
I'm going to give you probably triplets, triples, and then
Oh, God.
tell me. My bad. Okay, all right, let's do it. If you can't see the board, okay, I'll just, all right. What if I give you a DNA code that looks like this, okay? A DNA code that looks like this. I am reading right from the test, okay? So I would write it down. Some of you guys are still sitting there like, right? I'm like, I would write it down because I'm actually giving you a question on test. Maybe not the whole thing. I'm giving you a sequence.
Are we making this RNA or?
This is my DNA. Okay? Okay. GAG TTC. GAG TTC. Now that's my DNA. This is my DNA. Okay, now, what is the amino acid that's going to be coded to that? Now you got to do a couple of things first. Okay, you just can't take that and go right to there. Okay, because remember, what is a codon chart for? A codon. Codon is part of what? mRNA. Okay, so now we got to do something. We got to, one, take this sequence and make our messenger RNA from it. Okay, so what do? What's the rule of RNA? G to what? Write it down, write it down. Write it down. Okay. Alright. Now, we break it out into threes. Now look, a couple of key terms that maybe some of you guys are tripped up on, and that's why maybe some of you guys built your model kind of weird. Because you start not really understanding what they call a triplet. A triplet is this. That's in your DNA. Okay, this is called a codon. And then it goes to tRNA. Okay? Okay, that's an anti-codon. So now I'm using my codon chart. Okay, my codon chart, you take your 3 letters and you match it up with the amino acid that pairs up with the TNA. Alright, the tRNA right here. Take a look. What is the CUC? CUC. Well, obviously the last one doesn't even matter. Okay, leucine and what? Lysine. Alright, does everybody have any problems finding that?
No.
Seriously, raise your hand if you do, because I'll explain it. Alright, if you are having any problems. Now, I'm looking for 100 on this. Okay, nobody should get this one wrong.
So the tRNA is like the same thing as amino acids, or?
No. See that? If I was going to draw it, and she brings up a great point, a lot of people on the model messed it up, and the reason why, as a matter of fact, I think even the instructions are kind of weird on it because it made it look like you're looking up the tRNA. You're not. Okay, and then she brought it up to me, and it was kind of weird. It's like, okay, what you do is this. This clover-shaped molecule looks like this. Okay, has an end up here that binds with the correct amino acid that's sequenced to the codon. All right, I'll say that again. This tRNA, which is right here, has a anticodon down here. All right, so the anticodon for this would be what? EAG.
And then
UU. All right, now do you see why we had to go through that step?
the C at the end.
I'm sorry, C. Yes, it's actually the same thing as the DNA. Okay? So when you take a look, we had to take our DNA. Since it can't leave the nucleus, we have to make a copy of that and make a protein out of it. And the only way to do that is to go through this step and then to this step. Now, down here, you got the anticodon, and this pairs up with the messenger RNA, and on the other side of the molecule are the amino acid. So in order for the tRNA to, one, pair up with the correct amino acid, it has to, one, match up with the codon of the mRNA. Alright? So you got to think of it as a conglomeration, almost like a whole structure, because this guy now interacts with the ribosome that you guys constructed, and it produces a polypeptide and an amino acid sequence, which is a protein.
tRNA binds the amino acid before the mRNA, right?
That's correct. Yes. All right. Anybody got any questions? All right. Charles?
So the codon is what goes.
Yes, because this is called the codon chart.
Right. So it's not the ANS codon.
No. 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 Matter of fact, somebody built it, and somebody had a brother who was like a bio major. I think he was state or somewhere around here. And, it was all thrown off. And thank goodness he was there to help. Because, you definitely code by the mRNA. Now, the instructions is kind of weird, but what they were saying is that it pairs up here with the correct amino acid. They don't tell you where the amino acid comes from. It comes from the mRNA. It has to. That's the code. That's the codon.
So tRNA it only has one anticodon, right? So
Correct.
it's always going to bind the same mRNA codon. So it'll always carry on the same amino acid, right? Well, the anticodon, say the anticodon in tRNA is UUC.
Hold on. All right, I know. You know what you're doing? Okay, you're too smart. Now I want to say this is a compliment. The problem is we're not really staying focused on this. We're going off into way too advanced stuff. Let's just stay on this. And you're right. I don't want to confuse people, and I want to spend the next 40 minutes on one focusing on what's on this. Okay, and you're right. Okay, you start getting into that, and then somebody goes, "What the heck is he talking about?" Okay, and that throws off people. I don't want to do that.
So take a picture of that.
$20. All right.
Legit?
$20?
If you actually graft your eyes and do genetic mutation with an eagle, maybe you can see it, right? So then that could be put there. All right. What about, let's say, is there anybody has any questions? Right? All right. Okay, let's move on.
I actually
How about
have
this?
20
How about if
bucks.
I
I'm
give you
like,
these?
what?
How about if I give you this? Charles is like all bugged out, trying to take a look. What would be, let's say, the reverse? Can you actually go from, let's say, this to this? Can you actually do that? Hopefully you can, right? If I give you the tRNA, what would be the mRNA that would actually go with it? Okay, you could do that, right? On the test. Everybody should be able to do that. All right. How about, let's say, this is so easy. You guys are going to ace this test. Let me go over some questions. I'm reading verbatim. Listen carefully. The sequence of nucleotides in mRNA is determined by the sequence of nucleotides in what? DNA. Next. Which molecules contain uracil?
mRNA.
And
RNA.
good. Any RNA.
Wait, what about tRNA? mRNA,
You're recording it.
tRNA
It's like,
is transport.
I'm not gonna write this stuff
RNA.
down.
RNA is in the ribosome, and mRNA is the little guy that carries the message. Okay? How I actually think about this is that this is my blueprint. This is my encryption code to carry the message, and this is my translator. This is the guy who translates it to the ribosome. Okay? And that kind of is what happens. All right, let's move on. Let's see. If anybody gets this question wrong, they're going to go back to 8th grade. Ribosomes are the ribosomes of the cell are sites where the synthesis of what occurs?
Proteins.
That's easy.
Proteins.
What molecules contain cytosine? Both. Okay, how about, let's say, thymine?
DNA.
DNA. Thymine. Which ones contain thymine? Multiplicity. So on your essays, right, go back to multiplicity. Say, oh yeah, I know how to spell it now, right? Because I'm giving it to you. And I do that on purpose. So, all right, let's see. This gets a little more complex. This is actually maybe like a 9 or 10 out of 1 out of 10 as far as difficulty. During interphase, G1 and G2, what is synthesized? What kind of things are synthesized?
The proteins. The proteins and the copy of the DNA.
Proteins, macromolecules, right, which are large molecules that is necessary. What else? Certain RNA. Okay, certain RNA.
What was the question again?
Okay, during G1 and G2. Okay, there's certain things that a cell needs to do in order to divide and go through mitosis.
Right.
And there's certain things, the macromolecules that are necessary. I don't want to get into the chemistry of it. Certain proteins, like you said,
And then sugar, right?
Okay, and then certain ribose nucleic acids, certain RNA. Okay, needs to be replicated. Okay,
Would it be protein?
proteins are part of it. Yeah, without doubt.
Protein market.
Probably all of the above kind of answer.
Yes, thank you. All right, bless you. Let's see. Nobody should get this wrong, otherwise I will take back the diploma.
Multiple choice are there?
DNA is replicated in what phase of the cell cycle?
Us.
I gave that to you 10 times.
Wait, how many multiple choice questions are there?
30. 30 multiple choice.
I'm just so lazy. I'm just like, let's take this
Who's the
for
two scientists that came up with the DNA?
Crick. I need the crick
Who's
part.
the scientist that came up with X-ray chromatography?
Rosie something.
All right, now let's see who's really listening. What phase is the nerve cell? Okay, what phase is the nerve cell actually? Divide.
Because it doesn't divide.
It doesn't replicate.
You can incorporate that into your answer for your medicine if you want. Okay, it doesn't divide. And if we could somehow trigger
lazy.
that cell with what they call an initiator. Initiator means you send a signal to the cell to initiate getting past the R restricted system.
I saw like a documentary on stuff like that.
Shh, you gotta record this.
Just like biological, I don't know, they just cover like.
We'll do it later.
You could trigger. You could trigger something if you want to do it. Now, what's kind of cool, if you guys want to get into a neat field, besides genetics, okay? Think about it. Microtechnology. How about putting a little microchip that is like the size of, let's say, an atom, and putting it on there and sending it almost like a robotic arm, okay, to the axon and to send the signal, okay, almost like a miniature circuitry. Because what is a nerve cell? It's almost like a circuit. You have a axon that gets severed because somebody gets a sports injury, or let's say a car accident or whatever, and now it's not making a connection. So now what you want to do is maybe send some type of conductor to it, right? That'd be kind of neat to think about. Okay, that's the future. Incorporating maybe robotics into human body, or doing it biologically. Okay, that'd be cool. All right, anybody got any questions? All right, let's move on. All right, let's take a look at this question here.
I'd love to take a look at it.
What is the purpose of tRNA? If you were going to sum it up in scientific terms, what is the purpose of tRNA? Come on, somebody different. No, give me something that tRNA does. What does it do? No one's a smart man, listen.
Anticodon? Produces the anticodon.
Produces the anticodon, but what else does it do?
Transport it across the nucleus.
It doesn't produce it. What does it do?
It
It binds to that, right? And it also binds to mRNA to produce the anticodon, which synthesizes the amino acid in long term. Okay, that's correct. Okay, everyone understand that? That's probably where you can get tripped up, is all those words and everything else. All right, how about this? This is so easy, you guys are going to blow through it too quick. I should change it. I should make it harder.
You really shouldn't, because I'm freaking out.
You guys are too smart. You guys are too smart. Let's see. Once a cell passes through a restrictor site, what happens? What must it do? Yeah, it must divide. It must go through the mitosis stage, or the M stage. Okay? Once it passes through that restrictive point. What functions are served by cellular division? What actually you're trying to accomplish?
You want to get more cells.
You actually produce what? You're actually trying to produce two daughter cells, right? Is it actually different DNA, or the same exact DNA?
Same.
Exact same identical cell. So basically your parent cell and your daughter cells should be exactly the same. Because remember, it's happening in the S. There's no, unless there's a mutation. Okay, what kind of mutations do we have? And explain how they work.
You could have a amino acid added into the chain.
Okay, so let's do this. That's called an insertion. Okay? An insertion is now where we actually insert something here. Let's actually insert another G. Now you got a frame shift. We call it a frame shift, meaning that now everything gets shifted down. Does that make sense?
Yes, sir.
Okay, so now we'd have GAG, GTT, and now the C would be left alone. It wouldn't code. Okay, what about deletion? Instead of doing this, now we also get a frame shift, except now it's kicked up. Alright, does everybody see that? What's another type of mutation? And this is the one on the test.
A point.
Point, what does that mean? No?
It switches out the.
Good. Now, this is actually a very common one. Now, who's the proofreader? See who really read the book. Who's the proofreader that basically checks DNA polymerase? That's the proofreader. That's the guy that actually comes over and says, okay, we messed up. We got to fix it. That's nature's way of saying we got to fix it. All right? In the future, we're going to actually be able to do that and insert and delete certain genes in order to fix it. Okay, which is kind of cool. We're doing it now. All right, any questions? All right. Does anybody want me to go over anything? Because I'm almost done, and I think you guys are prepared as best as I can prepare you.
If you're done, why do you take this?
Yes.
Mutation.
No, and I'll tell you why. Okay, because the book is so terrible. They take
Book is a really
they
big
take all biology and they mix everything together. Yeah, sure. Viruses have DNA code in them, and they inject it into our cells. But if I was going to lecture about viruses, it'd be a whole different lecture, which I want to do. Because you should know about avian flu, influenza outbreak, right? All those cool things that have to do with viruses. You just can't stick it in the middle of genetics. It doesn't work.
So there's going to be nothing on the test
When
about...
people write books like that, it needs to be separated in bigger things. All right, this is called spiral education, where they take it and they try to do conceptualized spiral, and it's kind of, I don't know, a bunch of junk. Okay. I teach it a long time, and I know that you guys, if I just throw viruses in there, you're not getting the full thing. All right, anybody got any questions? Okay, let's take the rest of the period. If somebody wants to see me and go over any of the multiple choice questions or any of the essays, please let me know before we break again. Okay, excellent. I was hoping somebody would say that because I know some people are still messed up. Please listen. You're not excused yet. Listen. I hate to start a grade with a alarm. Okay, I just Googled this, and this is kind of the story here. Okay? We got our double helix. Okay, the first thing that happens is actually messenger RNA pairs up with my DNA, and it has to make a code of it. Okay, the only difference is that now, instead of the T, I'm replacing it with U. Okay? So the uracil is injected in here, so C to G and so forth, and the A now pairs up with U. Now, why do these things pair up? And this is an honors question. Okay, on my CP, the answer might be the shape, right, of the molecule. But why do they really pair up? Some of you guys are going to advanced chemistry. You got to know this. Okay?
Does that have to do with the hydrogen bonds and the color?
No, it actually has to do with the what? Come on, say people. Yeah.
With the number of bonds?
Number of bonds?
Yeah, like 2 or 3.
There's a shape of something that, now, the CP class, the answer on their test is the shape of the molecule, which I went over with them today. But you guys, I taught you 2 things, right? What are the 3 things that make up a DNA structure?
The
Phosphate, base sugar, and the bonds. Okay? Now the bonds go with each other. A to T, C to G. But why does an A go to a T? Okay.
Because one of them's a purine and one of them's other things.
One's a pyrimidine. Okay, purines and pyrimidines. Now one's a single sugar and one is a double sugar. And if you look at the chemistry, okay, that's how they have to bond. Now look, on this simple diagram, I gotta take this down because this gives you the answer. This is all the information for the essay. But you can see that the shape, but when you guys go to a basic I will have it blocked out. But when you can see that the shape of the molecule actually fits in, but it's more complex than that. You actually deal with the organic structure of how thymine looks together with adenine. And they actually bond together based on the chemistry. Okay? Alright.
That game.
Getting back to this.
So much fun.
Okay, guys, listen. We pair up the codon with the triplets. Alright, that's those two terms. Just know, these are the triplets here, every 3, and these are the codons. The mRNA sneaks out of the nucleus and goes to where?
Cytoplasm.
Cytoplasm.
Cytoplasm, and actually in the cytoplasm and the ribosome. Okay, it actually does because the DNA can't sneak out. It must stay in the nucleus. Okay, otherwise we wouldn't have to have RNA. So what happens is the mRNA sneaks out, it gets actually through the nuclear membrane, and then it goes to the ribosome. Now, in there, okay, right next to it, technically, the translation occurs. Okay, translation is where now mRNA pairs up with tRNA based on the codon and the anticodon, and that sends a signal saying, hey, look, I got a codon of GGU. That's glycine. Okay? That pairs up. Now, how does it really look? It looks like this. This is your amino acid here, this is your tRNA, and this is your anticodon here. The mRNA is going to be attached to the bottom. This is what you should have done for your project that you turned in. Alright, and we only had 6 tRNAs and also 6 amino acid sequences. Some people did them all and wrote them out, which is fine. But they only gave you 6. Now, the ribosome should be right next to it because that's feeding into the ribosome and spitting out what? Proteins. Alright, everybody got that? Alright, so this is really the answer here. Okay, so transcription is the information going to mRNA. Translation is messenger RNA teaming up with tRNA and translating that to make a protein. Okay? Alright. Any questions? Alright, that's it. You're done.